This is a production of Cornell University.
Thank you, Bill, for the introduction. And good afternoon, everyone. I thought that I would start with an introduction of the program that made the experience that I'll be telling you about possible. So this previous spring, I was a teaching assistant with the Cornell Prison Education Program, or CPEP, that's how I'll refer to it. It is a program that was founded at the university in 2013 and really motivated by the same founding principle of the university, that I would found an institution where any person can find instruction in any study. And to that end, they are dedicated to supporting incarcerated persons' academic ambitions and their preparation for successful reentry. And CPEP operates in facilities that are managed by the Department of Corrections and Community Supervision, or DOCS. That's how I'll be referring to it. Currently, they offer Associates of Arts degrees to men who are incarcerated at 4 correctional facilities. And they're shown here in green. All are within an hour's drive of main campus. And I wanted to emphasize just how humanities dominant the program is for the students. In a given student's program, there might be 5 STEM classes that they could take to get their associates. And unsurprisingly, plant pathology is not a staple course in their curriculum. It sort of depends on Bill's schedule. It is a medium security state prison for men capacity. It has just over 1,000 incarcerated individuals, or IIs, and that's the DOCS terminology. And in a given semester, there could be between 30 and 40 IIs who have applied for and are accepted and enrolled in CPEP. And also, unsurprisingly, there aren't a lot of publicly available pictures of the facility. So this is a Google Earth image that I put here to try to illustrate what it could be like to be there. You can see the wall around the grounds. This is the parking lot where, if you were a volunteer or a staff member, you would park. And you'd enter through this administrative building, go through a thorough security screening process, metal detector, get your prison ID, and then be escorted through a series of gates onto the grounds. And then as a volunteer, you are immediately escorted directly to the school building here, and this would be the only building you would see. Before starting as a teaching assistant, there were 2 trainings that I had to complete, one through DOCS and one through CPEP. And they're really designed to set the tone, really to scare you, one maybe more than the other, I think, as you'll see. But I wanted to share the most impactful lessons from the training before I get started with the rest. So first, the DOCS training. Upon entering the facility, you will be directed to pass through a metal detector. You will be hand stamped, and you may be subjected to additional search procedures. All items will be inspected. Matters of discipline are the responsibility of the security staff. Under no circumstances should you interfere. Do not bring into the facility any items that might be defined as contraband. Promoting prison contraband is a felony offense. This includes anything that is introduced with the intent to transfer to an II without the permission of the superintendent. And I later learned that it's not always a felony offense. It's at least a misdemeanor if you bring in prison contraband, but it can be a felony offense. And you will not be working with students. You will be working with IIs who are taking a class. Then the CPEP training. Prison is, to outsiders, a foreign country. It is wise to be quiet, observe, listen, learn the language, and identify the values of the prison culture. Don't be afraid to define yourself clearly. You are not required to conform to any stereotype IIs may have about you. Even after you have clearly defined yourself, expect to be tested again and again. Expect to meet many tremendous and valuable people in prison. Expect also to meet some champion manipulators. Do not be surprised if these sometimes turn out to be one and the same person. Prisons have proven that people do not learn very much from punishment, but more from example. What you do in prison is more important than what you say there. A broken promise to an incarcerated person is even more painful than in normal life, and it is an act of cruelty. And if after an initial exposure, you find yourself fearful of the prison environment, you should not continue to go into prisons. This work is not for everyone. So I was pretty apprehensive after the trainings, but I had already signed up to work with Bill. We had talked about the plans for the semester, and I was really reassured by the fact that Bill was a seasoned CPEP volunteer. So the class that I was a teaching assistant for was PLSCI 2225, Healthy Plants, Healthy Planet, Healthy People, taught by our very own Bill Fry. And there were 3 major learning outcomes for the class. First, that students would acquire a broad understanding of plant diseases, their pathogens, and how to manage them. To explain plant disease dynamics at the cellular, organismal, and population levels. And in the end, to illustrate their understanding and their knowledge in a term paper describing 5 important and diverse diseases on a crop of their choice, and how to manage them in an integrated way. And I also had a personal goal for this experience, and it was to determine what role I want teaching to play in my career. I'm in the last year of my graduate program, hopefully, and I've had some teaching experiences before at NC State and here at Cornell, but comparably, you know, in ideal environments with inexhaustible resources, and not really, but sort of, and professional students. And so I wanted a different representation of what teaching could be. So we met once a week on Tuesday mornings for a long and engaging 3-hour period from 7:50 to 10:50. Some days we could go in, like on Mondays, depending on cancellations or if we were going to do a study hall, but usually it was just once a week. It's hard to hold attention for 3 hours. Not necessarily a class that I would be excited to take. And I wanted to emphasize how limited of a classroom it was. So we had no PowerPoint. We had no internet, so no screens or computers or phones, and absolutely no samples. So no microscopes, no slides. You couldn't bring in plant samples. They couldn't even bring in rotten produce from the mess hall. There was nothing to point to other than our chalkboard and these 4 resources here. So we could bring in these printouts of PDFs, you know, conversions of PowerPoints as the lecture material. And we had these spiral bound readers as companions to the lecture material. They were filled with articles. Each student had a disease compendium for the crop on which they were writing their term papers. And then every student had a copy of the Agrios text. So this was our classroom. For me, on first glance, it doesn't really look too different from your regular sort of underfunded public school classroom. It kind of looks like where I went to high school. But there are bars on the windows here. And what you can't see here is a wall of windows for observation. But you can see the chalkboard and really the total lack of modern resources in a classroom. We had 5 students in the beginning, but 1 dropped out about halfway through the semester. And these were the 3 students who were comfortable with their photos being taken. So we had their permission. They were around between 30 to 60 years old. And outside of CPEP, they had no experience with college level education or agriculture. But I found that what they did have ample experience with was how frightening the prison environment can be. I can't speak to what it was like outside of the school building. We can imagine. But I found even in the school building it to be a very intimidating place to work, especially for a young woman. You can imagine. Um, Correctional officers have every power to question your motivations for volunteering and demand a response to that, to deny you entry, and to interrupt class with a canine unit and pull the students out and frisk them along the wall of windows, standing there watching you from the outside. So knowing that all of that was going to happen to them in the school building, they still registered for a class, let alone a plant pathology class. And most of the students said that they registered because they needed it to graduate, or they heard that it was challenging and they wanted to challenge. But there was one student, this student, who said that he registered for the class because his mom is a master gardener in Oklahoma, and he wanted to spend more time on his weekly phone calls with her, talking about what she was interested in. And he thought that our class could help him do that. So knowing all of this, Bill and I took an intentional approach to this semester. What we did first and very often was emphasize that everything that they will be learning is real. All of the ridiculous scientific names that we were asking them to memorize and spell correctly, and the diseases that are caused by those organisms, the economic losses that result from those diseases, that all of that is very, very real, and we can't show them, but we're asking them to trust us. We also used Phytophthora infestans as a teaching model. And this was in large part because of Bill's career-long expertise with the system. So we had a lot of material to pull from. But we also thought that if we stayed within one system to teach major concepts like Koch's postulates and life cycles, epidemiology, host resistance and management, that for people who were hearing it for the very first time, that might make more sense, especially then when we transition to talk about other organisms. And we also had them read the first 2 chapters of The Advance of the Fungi before we even met, which they all read. And if you're familiar with the text, it really sets the tone for Phytophthora infestans as a devastating pathogen, and I think helped sort of establish where we would be for this semester. We followed a sort of standard layout for each class over the course of this semester. We always began by answering what questions they had, ideally about the lecture material, but that wasn't always the case. And that could take a while, but we had 3 hours. So then we would review a list of important terms and summary notes, and Bill and I would compile the terms from the previous week and hand them out to the students the following week, just to make sure, to clarify, this is the information that we want you to know, and to make sure everyone had access to the same information. And these are just some examples of the summary notes. Then we spent the rest of the time discussing lecture material and a reading, if there was one. And I always ended with a debriefing session with Bill. And there are 2 sort of strategies that we utilized a lot in the semester that I wanted to share with you all. The first was that we sat in as much of a circle as we could. The classroom was small, so we couldn't quite arrange our desks and chairs that way, but we did it as best as we could, as this picture shows. And we would take turns going to the board. And so in many cases, no one was standing up front, and we were all in a circle, sort of equally participating in whatever discussion we were having. And if Bill went to the board, then I was asking questions of him as a student, trying to model for the others, these are the kinds of interactions that we are hoping that you have. These are the kinds of questions we hope you're thinking about. And in the end, I thought we were successful in getting a dynamic classroom where students appeared to feel very free to share when they were frustrated and confused, then excited, to help each other, to challenge each other, and to challenge us. Um, and and I think in the end, really, as I was hoping that this picture would show for you all, that we had a lot of fun, and that felt like one of the most important things that we could be doing. The other strategy that we used, well, I guess problem we were trying to address was how to give the students hands-on learning experiences in the absence of a lab. And what we ended up relying on was getting students to come to the chalkboard as much as possible to diagram like a life cycle, which is the case here, or to draw a disease progress curve. And the idea was that if a student were at the board, they were not solely responsible for diagramming whatever they were diagramming. They were relying on the people in the classroom to guide them through the process. So it was a low stakes environment for the person at the board and a mentoring learning opportunity for people in the classroom. And we came about this not because we thought of it on our own, but because in the second week of class, we had sort of voluntold a student to go to the board to diagram the P. infestans life cycle, which he did fairly begrudgingly and with some amount of pain. But then when he was on his way back to his seat, he stopped and turned to us and said, "That was the most I've learned in this class so far." And so we could come up with all of these, what we thought were innovative and creative ways to teach the information. But at least for that student, what mattered was going to the board and drawing something, and not to illustrate understanding, but to cement understanding. And I had mentioned that that after the course, Bill and I would have our debriefing session, and there wasn't an office for us in the prison, right? We couldn't stay. We were escorted out at the end of class. And so we went to our next best option, which was a Speedway gas station a couple of miles down the road, because it had this mini Dunkin' Donuts. And it had literally just 2 tables. And so every Tuesday late morning, this is where we would be debriefing about what we had experienced, mostly because it was jarring but meaningful. This is also where we would grade quizzes and come up with the summary term list for the next week. And an unlikely place for me to realize the sort of mentor that Bill is, and to develop a relationship with him. So at a gas station. So anyway, that was happening over the course of the semester. And then at the very end, we had the term papers due, and we asked the students to give presentations on the term papers. And we told them that we wanted lightning talks, 5 to 10 minutes, no more than 10 minutes. That should be sufficient for them to introduce the pathogen, the 5 pathogens on their crop, and how to manage them in an integrated way. And on average, each student took about 45 minutes to give their presentations, which we did not expect. So it took 2 classes to actually go through the 4 presentations. And we realized that this was probably, for many of the students, the first time that they gave a presentation to a respectful and attentive audience. So that was okay. And the student whose mother is a master gardener in Oklahoma wrote his term paper on the biology and management of peach tree diseases, because that is an important crop to his mom. And he wanted to connect with her throughout the project. And I was really touched by her involvement in his success in the class. He had her mail him these printed color photos of symptoms of the diseases that he was studying. And as in his paper. So not only was the student really invested in his success, but also his family. in contributing something as well. And so that was very impactful. I wanted to spend the rest of the time talking about some lessons that I learned from the experience and then close with some reflections from the students. And I wanted to say first that this was the single most impactful experience that I've had while in grad school. And I would encourage really anyone at Cornell because of CPAP here, but really especially graduate students to say yes to an unlikely teaching opportunity like this, despite the challenges. I learned the importance of trying to be precise and concise and to seek narrow, specific, and unambiguous words. And that this was especially important when the students you're teaching are learning it for the very first time and have little to no ability to check you on anything that you're saying. I also learned that I should admit often what I don't know and that I thought I knew things that I didn't. My ego got in the way very quickly in the classroom. In the beginning, I was very concerned with seeming like I was a good resource to the students, like I knew all the information really, really well, and found that I could be far more effective as an instructor to them if I were more honest than I was insecure. And so in light of that, I have a vulnerable anecdote about thinking that I knew things that I didn't, that I wanted to share. About a third of the way through the semester, Bill asked me if I would teach the students mitosis and meiosis, give a lecture on it. And I said, sure, I've studied subjects for years now that deal with some element of mitosis or meiosis. I feel comfortable giving a meaningful lecture to the students. And as I was spending the couple of weeks preparing for the lecture, I was thinking about what I would say are the core concepts of cell division that come to mind very quickly for me. And maybe that's how I'll inform the lecture. And that's what I'll share with the students. And one of those concepts was this notion I had that meiosis results in the production of gametes. And I thought that that would be a core concept I would teach them. And because we had used P. infestans as a model up to this point, I thought I would teach the subject through the P. infestans life cycle. So I got up to the board and I drew the scaffolding of this table, comparing and contrasting mitosis and meiosis. And I told the students I'm going to walk through the life cycle and at each stage talk about how mitosis or meiosis plays a role. And so that's what I did. But really, I distinguished the two here, saying that in this system, meiosis results in the production of gametes, and mitosis does not. And I thought that was concise and precise, and I think the students also thought that. And that was where we left it. And Bill didn't say anything. And then a couple of weeks later, Bill was introducing Ascomycete fungi to the class. And he was walking through the generalized life cycle, and he got to sexual reproduction. And he said something like, you have compatible hyphae that undergo plasmogamy, and you end up with multiple haploid nuclei in a cell. And those multiple haploid nuclei undergo rounds of mitosis. And then eventually, two haploid nuclei fuse. And very briefly, you have a diploid nucleus. And that nucleus undergoes meiosis. And then eventually there's another round of mitosis, and you get 4 sets of twins, your classic haploid 8 ascospores, and the life cycle continues. And I was nodding along and drafting my summary notes, like, yes, of course, this is what I've always learned. And then I was thinking about what I had been telling the students previously about mitosis and meiosis. And I was looking at this in my notes and looking at this, and I was like, wait, in Ascomycetes, meiosis does not result in the production of gametes. It happens in the zygote. And because meiosis happens in the zygote, the dominant nuclear state for the rest of the life cycle is haploid. And how fundamentally different that is from all organisms where meiosis occurs before parental nuclei are donated. And I was like, surely I knew this. Like, how did I get to this point in graduate school? And this is just now making sense to me. And like, it's now feeling very important to me. Why don't people talk about this all the time? This is so important. This is a level of difference that I didn't appreciate between organisms. And because the students had created this environment where it was not shameful to admit having not appreciated something and now appreciating something, I couldn't help but do the same. And so I hope I'm illustrating to you the dynamic in the classroom. It feels very vulnerable to admit this to a room of very real subject matter experts. But anyway, that was an experience I had where I admitted what I didn't know. I also learned that teaching added dimension to my research. I don't think that it hindered. I think that it only enhanced the experience for me. And there's another little anecdote I wanted to share related to this. So Bill had mentioned that for my research, part of what I do is I am characterizing diseases of rhubarb, primarily those that we think are the most common in the Northeast. And really that has led me to characterize a single fungal pathogen that causes leaf spots that look like this. And so this is, going on before I'm a TA and while I'm a TA. And maybe 2/3 through the semester, Bill asked me if I would teach the nematology lecture. And I agreed with some hesitation because I thought I should admit that I didn't really know anything of the subject to give a meaningful lecture to these students. So I spent a couple of weeks preparing. And while doing so, and then while giving the lecture, I realized that I found dagger nematodes and their ability to transmit nepoviruses to be really interesting. Like somehow I had ignored this in previous plant pathology classes. Like how did I not know that this was very cool? It's very cool to me now. And then at the same time, our lab started growing out a number of rhubarb accessions in the greenhouse that were previously in the field. So I had sort of closer eyes on them. And while they were growing out, I realized that in a single accession in our collection, there were these curious symptoms that initially resembled the leaf spot symptoms that I had seen before. And these are clones of each other, these two plants. And so I tried to isolate from these leaf spots the same pathogen that I know causes these leaf spots unsuccessfully. And I approached Mark Fuchs and asked him if he thought that any of these were worth screening for a number of viruses. In my experience, I have gone to Mark Fuchs many, many times with what I think are really cool symptoms, hoping that it's caused by some virus, and he always says, no, it's not worth testing. And so it was very exciting for me when he said, that's interesting, let's screen them. So we did, and what we found has resulted in a new project and a collaboration that I would have never thought that I would have as a graduate student studying fungal pathogens of vegetables. So we found tomato ring spot virus, which is a nepovirus, in this accession, causing or showing at least these symptoms. And that motivated a further collaboration with members of Mark's lab and a collaborator at an institution that I didn't think that I would be collaborating with, a high throughput sequencing of RNA project. And we have some interesting results. So our next step is validation. And I guess I'm not trying to say that it's because I was realizing that I was interested in nepoviruses as a TA that I happened to find a nepovirus in rhubarb. But it was that I had learned enough to be interested in the subject that it made something like this seem worthwhile last minute as an unplanned project. So that was something I learned. I also learned that any expectation I have of the students, I'll have of myself. So if I want the students, if I expect them to be interested in something, I have to demonstrate that I am interested in it. If I want them to spend time on something, I should demonstrate that I have spent time on it. I also learned that I should expect an experience to be interesting and meaningful, and then I may be better prepared to appreciate it when it likely will be. And I learned that I should be affected by the people I teach and to let them know that I'm affected by them. In a written reflection at the end of the semester, one student said, "I've never wanted to get out and bump into two people more in my life so I can shake their hands. Not only for what they did for me academically, but for treating me like a human being. I'll never forget them." So, it mattered what we were teaching them and how we were teaching them. But I really think that we could have been teaching them anything. It was that we cared about them that mattered the most. So some students' comments. "You started this class by telling us that everything we learn is real. I agree. Potato spindle tuber viroid. It's amazing that I know that. I would have never thought I'd say that before taking this class." And one student in talking about preparing for an exam, "Do you know that I can walk the yard with murderers and thieves and have less anxiety than I do in this class?" It's really not what we meant to have happen. "I'm using the disease triangle to explain recidivism with my reentry program mentees. They came here because of some interaction between their behavior, their needs, and their support system. They have to change at least one to keep from coming back. I will never look at a piece of fruit, vegetable, or food the same way again, knowing that a grower's many efforts somewhere along the line have made that resource available to me. And I would love to show my family this." I'm happy to report that a number of the students in our class graduated this semester. And those who are still incarcerated will continue taking classes through CPEP as long as they're offered. I care a lot about this experience. And I really, really appreciate the people in this picture with me, especially Bill, for making this possible. And you all for your attention. And I'm happy to take any questions.
There's some questions for Libby. Get us started. I have one. How would you compare your Cornell undergrads with
Oh.
CCF undergrads?
So how would I compare the students in the CPEP program with students I've taught at Cornell? There is a major, major difference in the background education with the students at Cornell versus students at CPEP. It's not surprising. The understanding of plant science, of DNA, of biology broadly, that was a surprise for me. The students at CPEP did not have a strong understanding. But they had almost no desire, I think, to prove themselves to each other, perhaps in the way that I have observed with undergrads here. What I had found really meaningful about the experience is something I had talked about, was that the environment in the classroom was very free and willing to admit when you didn't understand something. And I think as a TA in Cornell classes, would have been very scared to say to anyone that I had just appreciated that meiosis occurs in the zygote in Ascomycetes. But that was totally welcome in a CPEP classroom.
Is there a prison program for women?
So if there's a prison program for women, my understanding is that CPEP currently does not operate in women's prisons, and I don't know if there is a reason for that in addition to maybe they are too far away for people to volunteer at. Yeah, I don't know where the nearest women's prison is, but there are other universities have prison education programs at women's prisons. In New York State, Brown operates in some. Columbia has a program, but not Cornell.
First, great talk. And I want to follow up on Bill's question, actually comparing the students. So, if I understand correctly, these students in the prison are not really having the type of background that's really needed to go into advanced topics, and you're actually teaching them pretty like specific terms and terminologies and stuff like that. How, in your observation, do these students react to like the frustration compared to other like Cornell undergrads who have more backgrounds and stuff? And especially like, are there any incidents where their frustration could like lead to like stopping the class or getting the guards involved?
Okay. Yes. So Igor, your question was about comparing perhaps how students react in CPEP versus Cornell to not understanding a concept, and if their frustration ever elevated a situation to a point of concern. I would say that there were many times where students were frustrated by not understanding a subject, but we would just slow the class down. And with 5 students, and eventually then 4, it was very easy to make sure that everyone was on the same page. They often turned to each other for support, so they would tutor each other outside of the class if they had recognized that someone wasn't understanding something. There was never any concern that I had about safety in the classroom once I felt comfortable, not at first, but eventually after I learned the students and knew who they were. But they handled frustration with the subject, I think, similar to how I would have expected Cornell students to. Yes, Kathy.
I noticed that some of your early summary notes had graphs in them, like area under the disease progress curve. And I'm thinking of like my magical mushroom students. And if I put up a graph, I have to be like, okay, like, like how did you explain the area under the disease progress curve?
How did we explain? Yeah, the charts and figures. There was a lot of repetition at the chalkboard and getting the students to draw it out themselves.
You got it?
I think so. Some. Yeah.
Tough one.
Yeah. There were examples where I mean, we spent maybe 2 weeks on disease progress curves and exponential growth and trying to compare exponential growth to maybe how they might have thought about their finances before and what happens in a savings account, and that maybe wasn't as helpful as I thought it would be, but walking them through individual calculations, getting them to sort of debate each other on what they thought the outcome would be, and then and argue their case. Yeah, we did that.
And can I say one more thing? Like, I really liked your reflection on intellectual humility.
Hmm.
You want to say that into the mic for the Zoom folks?
Okay?
I'll let you have the floor. I think this one is on.
I'm not giving this seminar, but I wanted to say, this is Kathy Hodge. I wanted to say that I really liked your reflection on intellectual humility. And I think it's a really good attribute to have, right? And when you think about it, like we're all just learning all the time. And in fact, that's like why we're here, right? We're curious about the things we don't know. But I think when students first graduate, often with a PhD, I think you feel like you should be an expert on things. And I still feel like I'm supposed to be an expert on things, but in fact, I got here for the very opposite reason that I didn't know anything and I just want to learn more and more stuff. And I just think that intellectual humility, like you've shown, is just a beautiful way to like let people in and just recruit them to the process of science. You know, like there's so much we don't know. And so if somebody asks you a question that you don't know the answer to, it's like, that's a beautiful moment if you can say, that's a really good question. And, you know, like, I don't really know. So good job. Want to say anything?
That was lovely. I don't know if I can follow up. But that was, I think, very much the dynamic that I experienced in the classroom.
So-
Yes.
I wanted to say one thing. When Libby first started, I really did not know that you were apprehensive about going into the prison.
Right.
I guess I'm not very observant. Could you compare just describe your experience early on with that later on? And maybe we could stop with that one.
Okay. Yeah, so in the first month or so of the semester, I tried really hard to hide this from you. I guess it worked. But I didn't really sleep the nights before going in. And I was very afraid of what version of a correctional officer I was going to meet at the gate. I had heard sort of horror stories about previous volunteers. This didn't happen to me, but you know, your shirt was too tight, so you can't come in today. Or you're wearing a white shirt and it's going to rain, so you can't come in today. Or are you sure you're a volunteer because we don't have your name on the list, even though you've been there for weeks, and they need to have someone come over and look at your, you know, identifying information to make sure that you can go back. And so I was really scared simply to enter the facility. I remember the first time that you took a bathroom break in lecture and I was sitting alone with the students, and it was our first class and I didn't know anything about them and they didn't know anything about me. And I was so afraid that all they could see was that I was young and a woman. And I was very wrong about that in the end. By the end, I think somehow we achieved like another sort of classroom dimension that didn't exist in the prison. Like you'd walk through the prison, but get to class and it was a classroom, and that was awesome. And it could have been anywhere. So it very much changed by the end. Yeah.
Okay, Libby, thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.